And we've got a fantastic speaker, and the description that you shared is, how do I follow that? Really? So Daniela Zárate, did I get it right? I hope so, is a PhD candidate in UC San Diego's Division of Biological Scientists. She works out of Professor Joshua Cohn's laboratory in the section of Ecology, Behavior, and Evolution. Daniela's dissertation research takes an interdisciplinary approach to studying the Western honeybee, the world's most important agricultural pollinator. Her research features a comprehensive assessment of genomic ancestry in honeybees from across a broad geographic expanse, as well as a behavioral study of aggression of Western honeybees on both the individual and colony level. And beyond that, she uses her platform to challenge institutional oppression of marginalized communities and decolonize academic and scientific spheres of influence. She is a prison abolitionist and is involved in efforts to bridge the institutions of education and mass incarceration. She enjoys being part of the Biota Project, a collective of scientists and artists interested in using mixed media art for science communication and social justice. I'm really looking forward to hearing from you. So welcome, Daniela, please. Thanks, everybody. Great talk.
Oh my God, hello, my beautiful people. It's so good to be here. I am so excited to be here. Thank you so much, Michael. Thank you so much, Emma, all the museum crew and staff who are made this event possible. And thank you all today for being here. This means so much to me. I can't even begin to tell you. I am San Diego born and bred. Is anyone else San Diego born and bred here? Yes, San Diego born and bred. I grew up in Escondido. I went to this museum my entire childhood. And so I mean, I remember when this museum, you could go into the Airstream and smooch your sweetheart inside there. You can't do that anymore. You blocked it off. So, but I remember the good old days, okay? And so I am so excited to be here and to talk about my research. I dressed up for the occasion. I put on my bee suit just to be in the spirit of things. And again, thank you so much for the amazing introduction. This means the world to me to be here. And I'm just excited to talk about honeybees. So let's get into it. Let's move my slides. All right. Hopefully this will work here. Okay. Doesn't want to advance. Let's see. Is this on? Let's see. Can we? Yes, please. Thank you, Michael.
Oh, something just advanced.
Oh, okay, there we go. Perfect. Thank you so much. Thank you so much. Okay. So I am currently getting my PhD at UC San Diego. I'm a few months out from graduating and moving on to another system. So what I'm going to be talking about today is my dissertation research, which I basically do 23andMe on honeybees. So in case you don't know what 23andMe is, it's a genetic ancestry analysis service. Anyone with $100 in their pocket can go out and find out what they're made of. And before we get into that, I just want to briefly say that I would describe myself as a biologist who is interested in social insects, honeybees, ants, anything that lives colonially in a big hive. I want to study it. I want to research it. And honeybees, when I first was giving this talk to Emma, she asked me, how did you get into honeybees? And honestly, I have to tell you that I never thought I would ever study honeybees. I was always someone that was in a rat lab or in a mouse lab. Even I spent a few years in a plant lab. I never thought I would ever be studying honeybees. But when I came to UC San Diego, my PI, my principal investigator, he had a research project on honeybees. And he asked me, would you be interested in studying honeybees? And I was like, okay, yeah, that sounds cool. And I never looked back. 7 years later, I'm still working with honeybees. They're beautiful creatures. I took some of this photography myself here when I was working with some of the honeybee hives. They're gorgeous creatures and I fell in love with them. I want to study them the rest of my life. And so I'm really excited to share my work with honeybees with you today. Something else that I'm interested in besides honeybees is this evolutionary concept known as genomic admixture. So admixture, maybe you haven't heard of it before. Basically, admixture is the mixture of genetically distinct lineages. So what does that mean? Genetically distinct lineages can mean a variety of things. It could be species mixture like grizzly bears and polar bears. It could mean the individual admixture between an individual of a species with another individual of the same species. It could be subspecies breeding like wolves and dogs, which are different subspecies of each other. So genetically distinct lineages can mean a variety of things on several different levels. And the admixture that we're probably most familiar with is the admixture that makes you, you. So when one lovely romantic evening, your parents decided to have you, the combined two distinct genetic lineages, your mother's lineage and your father's lineage, to make you. And the sperm and the egg came together and they formed a zygote that eventually reproduced into several different cells and it became you. So you are a product of two distinct genetic lineages from two distinct individuals, and it makes a family. So that is what a family is genetically distinct lineages mixing together. And I want to talk a little bit about my own genetic admixture. So I'm Mexican-American, and my mom is from this super colorful town in Querétaro known as San Joaquin, and very, very colorful town. And my father was actually from a state in southern Mexico known as Oaxaca. And together we created a family that's as colorful as the one that you see in Encanto. We all have our superhero powers. Mine is obviously a bee scientist. That's my superpower. And so genetic admixture is this beautiful evolutionary phenomena that can create magic. And if there's one thing that you take away from this talk today, it is that admixture is a common and widespread biological phenomena. A variety of organisms across taxonomic levels participate in admixture, from howler monkeys to toads to zebras to wolves. A lot of things like to interbreed across species and subspecies lines. And sometimes that interbreeding isn't the best thing, like the horse and the donkey together breed to create a mule, which some of you might know is actually a sterile creature. It's not able to reproduce, can't have babies, very, very sad. That's a result of what we call genetic incompatibilities. Basically, the DNA of different species sometimes doesn't play well together, and not all love stories are meant to be. Offspring aren't created from all mixtures. But admixture can also be a creative evolutionary force. And actually, modern Tibetans hold in their genomes signatures of ancient admixture events with other hominid species, the Denisovans, that help them stay adapted to high altitude conditions. Sunflowers also like to cross across species lines, and their offspring are able to colonize and thrive in extreme environments such as dunes in the desert, which the parent species weren't able to colonize, but their hybrid, admixed, intercrossed offspring can. Also, here's one for the Aquarius lovers here. I have 2 freshwater tanks filled with cichlids at home, and cichlids are actually the biodiverse explosion of cichlids that we have today are the result of admixture events that created the amazing biodiversity of cichlid fish that we see today, all thanks to cichlids one day deciding to cross and interbreed across species lines. And bees also like to interbreed across subspecies lines. But before we get into that love story, let me say that if there's another thing that you take away from this talk, it's that our beloved honeybee is not native to the American continents. It's actually an invasive species. It's native to the continent of Africa and Europe and near Asia. Everywhere in that map that you see blue, it's where the honeybee is native to. Everywhere else in the world, it's been introduced thanks to human activity. That's not to say that the Americas didn't have their own bee species. Actually, the Americas had a wide variety of different bee species. This is an image from a Mayan codex showing beekeeping practices and bee gods. The Americas actually had various species of stingless bees that were very popular, and the Mayans were so enamored by their stingless bees that they had a whole pantheon of gods that they worshipped for beekeeping. But let's talk a little bit about the Western honeybee, our beloved pollinator, the one that everyone loves. So another thing that you should take away from this talk is that the honeybee, the Western honeybee, the one that we're all familiar with, is separated into 4 major biogeographic clades. We call them the big 4 honeybee clades. Those are the bees from Africa, the bees from Western Europe, the bees from Eastern Europe, and the bees from the Middle East. There are a couple of other groups of honeybees as well, but we're not going to focus on those for this talk. We'll just focus on the big 4 honeybee clades, or the big 4 honeybee groups. Another thing that you should know is that honeybees are taxonomically complex. There's over 30 different kinds of subspecies of honeybee, which was amazing. That blew my mind when I first heard that. I always thought there was just one honeybee and that was it. But apparently there's tons of honeybee species, subspecies. And this graph here is just an image of a phylogenetic tree, an evolutionary relationship figure showing how the different bees are related. And we have the African bees, the Western European bees, the Eastern European bees, and the Middle Eastern bees. And I bet a bunch of you are like, they all look the same, right? But they have subtle differences in and how they look, and also even bigger differences in how they behave. So the actually this bee right here, it's Apis mellifera carnica, is known as the Carnolian bee. They're very blonde, they're very yellow, very gentle, whereas Syriaca is a little bit darker. It's a little bit more defensive when you open up its hive and its colony. So actually, let's go back here. So when the Americas were first being discovered and people started coming over to the Americas, they started bringing over honeybees because they wanted to have their source of honey with them at all times. So they brought over a bunch of Western European and Eastern European honeybees. So we've had those bees here in the Americas since the 1500s. However, bees from Africa weren't brought over to the American continents until 1957, when some scientists decided to bring over some African queens for experimental breeding purposes. And in 1957, they brought some over into Brazil. And what ended up happening was that those bees escaped out of their apiaries and spread across the continents in an unprecedented, unexpected, spectacular biological invasion, and took over the American continents. And along the way, these African bees interbred with the pre-existing European honeybees that had existed in the American continents since the 1500s. And what ended up was the result of that was the so-called Africanized honeybee, also known as the killer bee, because it is a little bit defensive when you open up its hives. So the killer bee. That's how the killer bee came to be. And I just want to take a pause here actually and make a note about this idea, this concept of the Africanized honeybee, because when I first started studying honeybees, I always called it the Africanized honeybee. Other people called it the Africanized honeybee. Everyone called it the Africanized honeybee. But the more I started learning about honeybees and Africanization, I started kind of questioning about whether that was the right thing to call them, because The African honeybees are a diverse group of over 24 honeybee subspecies, and only one subspecies was introduced into Brazil in 1957. And so to call the Africanized honeybee such a generic name as the Africanized honeybee, when you only have one subspecies really representing the Africanized honeybee, it was a little bit, I think, it was a disservice to the biodiversity of African honeybees. And so I really think we should start rethinking calling the Africanized honeybee the Africanized honeybee. But that's just some food for thought that I want to leave you there with. So my dissertation research was largely inspired by this question of, I wanted to study and learn about genetics and behavior in the Africanized honeybee, or the AHB as I call it. And the first thing that I want to know was, what is the genetic composition of the honeybees in the Americas? So this is the 23andMe question. And some researchers had conducted some studies about this. The study that looked at honeybee admixture in Brazil showed that honeybees were 75% African, with the rest being from the Western European lineage. And here in this graph of Brazil, or this figure of Brazil, each individual pie chart is one honeybee genome, and the colors correspond to the various honeybee clades. So as you can see here, these genomes, these pie charts are mostly pink, with the rest of it being black, meaning that they're mostly African. And then the bees of Uruguay are mostly yellow, and that means that they're mostly Eastern European in origin. So in Uruguay, we have about 75% coming from the Eastern European honeybee lineage, and the rest from the Western European. A little bit closer to home, what do honeybees look like here closer to home? Some researchers in Texas and New Mexico did a study, and they showed that before Africanized honeybees reached the United States, they were pretty European in nature. They were mostly Eastern European and Western European. But after Africanized bees reached Texas, their genomes turned mostly African. So it's mostly pink here, with the rest being from the Western European lineage, and the same in Arizona as well. So my dissertation research then wanted to ask, what is genetic ancestry in honeybees from different regions in the Americas? Because those two studies I just showed you looked in Brazil and they looked in Texas. And that's a lot of space between two data points. So I wanted to see honeybee admixture in other areas besides those two. And that's where I basically came in and did 23andMe, but on bees. And you have to give me some license here. Bees actually have 16 chromosomes, not 23, but you have to give me that little bit of poetic license. It just rhymes, it just fits so perfectly. So, okay, so I had three major questions when it came to honeybee genetics. The first was, what is the genomic ancestry of Africanized honeybees? The second was, how does that compare to mitochondrial ancestry in honeybees? And the third question was, what about the genetic diversity of honeybees? And it's okay if you don't know what these terms are, we're gonna go over them right now. So Bio 101, let's talk about the cell. This is a cell here. Cells have a bunch of little organelles in them. And I want to point out the nucleus here. This is the control center of the cell. That's where most of our DNA exists. That's what we think about when we think about 23andMe and genetic ancestry, the DNA in there. However, we also have the mitochondrion. This is another organelle. Some people like to call it the powerhouse of the cell. Yes, exactly. People call it the powerhouse of the cell. It's where we get all our energy from in the cell. And the nucleus has DNA. It has most of the DNA in our cells. And so again, this is what we think about when we think about 23andMe. You're thinking about the DNA in your nucleus. And your DNA in your nucleus comes from your mom and your dad. So pretty basic, pretty straightforward. Most of us kind of know about this, are familiar with it. This DNA is from your mom and your dad. However, the mitochondria also has DNA. So that's something a lot of people don't know. The mitochondria also has its own DNA. Why does it have its own DNA? Well, because actually back before, human cells were a thing and cellular evolution was happening, the mitochondria actually came from bacteria, like our protocells engulfed bacteria that eventually became the mitochondria. And that's why mitochondria has its own DNA. It also has its DNA. But what's interesting about mitochondrial DNA is that it comes only from your mother. So your nucleus has DNA, it comes from your mom and your dad. Your mitochondria has DNA, your mitochondria only comes from your mother. And so that's why people like to call it, like to think about this term called mitochondrial Eve, because if you think about it, if your mitochondrial DNA came only from your mother and from her mother before her and her mother before her, it all is coming from one maternal source. So that's why I was interested in mitochondrial DNA and nuclear DNA, because they're inherited very differently. And so you're able to think about two very different forms of inheritance, of genetic inheritance. I can look at the maternal lineage through the mitochondria and both the maternal and the paternal lineage through the nuclear DNA. I also wanted to think about and measure genetic diversity in honeybee populations. So genetic diversity is variation. It's what makes maize super colorful. It's honestly what creates the richness of biodiversity that we see in the world today. This is all thanks to genetic diversity. It's something that conservation biologists are always really interested in because they want to conserve genetic variation and genetic diversity. So I wanted to see how diverse are honeybees in their genomes. And actually humans also are we're able to map genetic diversity in humans as well. So the most genetically diverse humans are in sub-Saharan Africa, the cradle of civilization, and the least diverse in East Asia, probably due to a population migration bottleneck there. So how did I do this? Well, I had to collect Africanized honeybees from various ranges in the Americas. So I was able to collect 15 honeybees from San Diego, California, right at home. I was also able to collect from Chiapas, Mexico, which is a state in southern Mexico. I also was able to collect in a national park in Costa Rica and in Barra Colorado Island in Panama. So I collected from 4 different sites, 15 honeybees for a total set of 60 Africanized honeybee genomes. And I actually, when I say in situ, that means it's a field term, like field ecologists use this term to say that it's a fancy term to say that you went around the rainforest and you caught some honeybees with a net. And I actually did collect honeybees with a net walking around in Panama and San Diego County. It was a lot of fun. I had a lot of fun doing it. So that is what in situ sampling is. And this is just a map showing where I collected in San Diego County because we're all familiar with San Diego County. I collected all over San Diego County. I collected in La Jolla. I collected in Ramona. I collected in Coronado. And actually, my brother is here today, helped me actually collect some of these honeybees one day, and which was amazing because he's allergic to honeybees. So that was a lot of fun. Okay, and then I had 60 honeybees, okay? I had 60 Africanized honeybee genomes. I had their physical bodies and test tubes in the lab. Now I had to collect honeybees to compare those Africanized honeybees too. So when you have a mixed genome, you want to have pure genomes to compare that admixed genome to. You need an ancestry reference panel. So how I collected those pure lineage honeybees was, thankfully, in science, people like to sequence a lot of DNA. And so there's databases available online that you can go ahead and download whole honeybee genomes from. And so thankfully, there were 2 science teams that had already gone ahead and sequenced a bunch of African, Western European, Eastern European, Middle Eastern honeybee genomes. And so those were all online, and all I had to do was go onto the database, the National Center for Biotech Information. I just had to go on there and download 99 genomes, which was a lot of fun. So I downloaded 21 African genomes. I didn't go to Africa to collect African honeybee genomes. I just went onto a database, downloaded some genomes, 21 African genomes, 29 Western European genomes, 29 Eastern European genomes, and 20 Middle Eastern honeybee genomes to give me a grand total of 99 ancestral honeybee pure genomes I could compare my Africanized honeybee genomes to. And I downloaded different subspecies. So these are all the subspecies I downloaded, which wasn't as exciting as going out to Panama and collecting genomes, but also a lot of fun. So at the end, I had 159 honeybee genomes that I was working with, 60 that I had collected myself, and 99 that I had downloaded from online databases. Oops. And then what do you do with those 60 physical bodies? Well, I did my best Queen of Hearts impersonation, and I cut off honeybee heads to extract DNA from and send off to sequencing. So after I extracted DNA from honeybee heads, I sent off to the UC San Diego's Institute for Genomic Medicine to sequence some honeybee genomes. And I got back lots of DNA. And if you've ever worked with DNA, DNA can be huge. I have a normal laptop, and I probably can only open 2 DNA sequences on it before my laptop crashes because it doesn't have memory. So DNA can be huge. And because it's so huge, oh, and funny thing. So before this, I worked in a rat lab, right? And I was always on the bench like these wonderful scientists here. And I feel like that's what my friends think I do when I'm in the lab. And this was my first time actually becoming a computational scientist and working almost exclusively in the in programming and computer coding. And when you work with DNA, all you do is computer program, basically. So I want to actually thank Jude Poole, who taught me how to be a computer programmer and analyze DNA sequences. He's director of IT at UC San Diego. I also want to thank Aaron Calfee at UC Davis, who helped me a lot with my analyses. And the San Diego Supercomputer Center, I want to thank them as well. They helped me analyze a lot of my DNA. So when you work with huge data sets, you kind of have to work on supercomputer clusters because there's no way your normal laptop is going to open up more than one DNA sequence. So thankfully at UC San Diego, we have a nice supercomputer cluster, and it has a cool, huge cluster that I'm able to access pretty easily. And I basically like to tell people that I'm Neo and I work in the matrix because that's what it feels like when I'm on my terminal, just working with my DNA sequences. And actually, this is all fun and games when I say it, but there's times when I'm working on DNA sequences, I'm just like in the matrix. And one time I was trying to figure something out. I was trying to analyze some sequence and it wasn't working. And I was so frustrated. And my friend Jude Poole, the director of IT, who's always helping me with computational stuff, he turns to me and he says, maybe your problem is cosmic rays. And I was like, what? Cosmic rays? They told me that grad school was going to be hard, but they did not tell me it was going to be this hard. But apparently cosmic rays sometimes flip binary code when you're coding, and you have to protect against cosmic rays. Tech companies actually have to protect against cosmic rays flipping a binary digit in your code. And so Jude was telling me maybe it's cosmic rays, and I almost quit that day. Like, I could not. It was oh my goodness. But thankfully we analyzed it. We figured out what it was. It was not cosmic rays. It was something else. But that's just goes to show how computational biology can sometimes be really extra. But so anyways, so now I have lots of DNA, right, back from my honeybee sequences. And the first thing I wanted to do was assess population structure. So I just wanted to get a big picture of what does population structure in my 60 honeybees sampled from my 4 countries look like. To do this, I did a principal component analysis. So principal component analysis is a really fancy term. It's a multidimensional data reduction technique used to simplify complex data sets. It's able to take millions of genetic markers and just kind of draw out big patterns within that data. I'm going to show you an example of human genomes mapped in principal component analysis space. Just kind of look at it. Don't try to analyze it. Just kind of see what emerges out of the pattern, okay? Okay, so here's a principal component analysis figure of human genomes. And you see that each individual data point is a human genome, and they're kind of grouped by region. So we have our sub-Saharan African genomes down here, our European, North African, and Indian genomes up here, and lastly, our Asian genomes from various regions up there. And the biggest thing that should emerge out of this is that, you know, some are closer to each other and some are further apart from each other. And that's all you should get from this principal component analysis. So just kind of a big picture, what do things look like? Can I start to see patterns in the data? Now I'm going to show you what my honeybee genomes look like in principal component analysis space. All right, so there we go. Okay, let's start with the Western European honeybee genomes, kind of sitting up there in the corner. We're moving on to our Western European genomes, our Middle Eastern honeybees from Syria and Jordan. Lastly, our African genomes. They're all kind of spread out in principal component space. Let's start looking at where my Africanized honeybee samples. Our Mexican samples are kind of at the bottom there, next to the African group. The Costa Rican samples right below it. The Panamanian samples right below those samples, all kind of clustered near the African genomes. And our San Diego samples right in the middle of all four ancestral groups. And so we start to kind of see relationships between the four countries there. So the next thing I wanted to do was the 23andMe part. I wanted to know what exactly genetic ancestry-wise are my honeybees. So to do this, I did a genetic ancestry analysis, 23andMe. So, okay, this is going to be a lot of data, so stay with me. I'm going to show you on the left side, Old World Honeybee Reference Panel. So these are the 99 honeybees I downloaded that are pure honeybees, okay? Ancestry proportion is going to be on your y-axis, and here at the right-hand side, we're going to have my Africanized honeybees. Okay, I'm going to show you piece by piece. So here are the African genomes, and the colors correspond to the different major honeybee clades. And so each column is one individual honeybee. We have 21 of these honeybees here, and they're all red, right? Like there's a little blue up there, but mostly they're all red, meaning that 99.9% of their genome is African. So that makes sense, right? I downloaded these honeybees. They had the label that they were pure African honeybees. So in the ancestry analysis, they came up pure African honeybees. Okay, moving on to the Western European honeybee genomes. Again, black is the color for Western European. So these honeybee genomes, again, mostly black. So we have some, you know, admixture going on here, some weirdness going on, but mostly they're all black, meaning most of their genome is Western European. Eastern European, represented by the color yellow, most of the genomes here are, again, almost 100% Eastern European. Then we have our Middle Eastern genomes, bees from Syria and Jordan, and that they are represented by the color teal. So those are our four major honeybee groups. We do have a little bit of the Syrian bees are showing some African ancestry, which just means that whoever collected those bees collected bees that were mixed between Syrian honeybees and African honeybees. But mostly their whole genome is pretty Middle Eastern. Okay, now we're going to move on to our Africanized honeybees. So let's start off with our hometown, San Diego. So now you can see that the mixture is a little bit different. Instead of having individuals that are, you know, purely one color, we have a mix of colors. We have about 40% of San Diego honeybees. A honeybee in San Diego is 40% African, and then about 25% Eastern European, and about 20% Western European, and about 8% Middle Eastern. So let's move on to Mexico. The bees in Chiapas, Mexico are about 70% African and about 18% Western European. and maybe like 4% Eastern European. The bees in Costa Rica are about 85% African, with the rest coming from the Western European lineage. And then lastly, the bees from Panama are the most African of all. They're about 95% African. So that is the 23andMe of Africanized honeybees. And whenever I see this data, I always think about Jackson Pollock. And if that's all you see right now, I understand, because I look at my data kind of like how I feel like an artist looks at a Jackson Pollock. And I actually don't really like Jackson Pollock, but but some people don't like data. So there's that. So, okay, so I think one of the biggest takeaways from that 23andMe analysis is that San Diego honeybees are pretty diverse. We have very diverse honeybees here. Our honeybees have 4 different major honeybee lineages in them, which, you know, that is amazing. Our honeybees are pretty cosmopolitan, I would say. And why would San Diego have such diverse honeybees? And honestly, maybe because San Diego is the greatest city in the world. I think that is the answer to it. I think that's the only answer. But no, there's various reasons why the San Diego honeybees are pretty diverse. And, you know, some people don't even know why. And I still also don't fully know why we have such diverse honeybees. There's lots of reasons for that. One reason is that here in the United States, we tend to work with European honeybees, and compared to Mexico, Costa Rica, Panama, those countries don't work with exclusively European honeybees. We actually work with Africanized honeybees. They don't try to requeen their honeybees. They don't try to exclusively breed European honeybees like we do here in the United States. Those countries have just embraced that the Africanized honeybee is here to stay. Whereas here in the United States, we actually really try to maintain our European honeybees because they're easier to manage, they're more gentle, they like to make honey, things like that. So the fact that we're maintaining all these European lineages is probably one of the reasons why in San Diego we have such diverse honeybee lineages. Okay, so the second question now. How does that compare to the mitochondrial ancestry of Africanized honeybees? So now we're talking about that, again, the mitochondrial DNA inherited from the mother. And to do that, I did a phylogenetic analysis. So phylogenetic analysis is an analysis where you group organisms together based on evolutionary lineages. A really nice example of that is the evolution of Eevee, the Pokemon. Like if you look at this diagram here, you see that Eevee evolves into various lineages. You kind of see how they're related to each other. I found this example of a phylogeny online, SpongeBob phylogeny, showing like sponges here with SpongeBob porifera up to the mollusks. So phylogenies are basically analyses of evolutionary relationships across organisms. So this is my phylogeny with my 159 honeybees on it. Don't squint at it, just kind of look at it. I gave you a table here that you can look at the numbers of mitochondrial ancestry. So in San Diego, 60% of the honeybees have African mitochondria, 13% have them from the Western European, 0% have them from the Eastern European, and 27% from the Middle Eastern. Mexico, 73% of them have African mitochondria, 27% have Eastern European mitochondria. Costa Rica and Panama, 100% African mitochondria. So from this analysis, I deduced that African mitochondria is pretty prevalent and predominant in the Mexican and Central American samples. And in San Diego, we continue to see this diverse representation of honeybee genetic material. Okay, so last question that I wanted to talk about was how genetically diverse are honeybees? To do this, I did a genetic sequence similarity analysis, which when you want to measure genetic diversity, you measure this thing called pi, which is the mean number of nucleotide differences per site between all possible pairs of sequences in a population. So basically, how different are DNA sequences across individuals in a population? So I'm going to show you a graph of genetic diversity. starting with the African honeybees. So they're about 0.0045 in terms of genetic diversity. The numbers don't really matter, just the ranking, the similarities, and how things are relative to each other matter. So the African is pretty diverse. Western European bees, not too diverse. Eastern European bees, even less diverse than Western European bees. And actually, it's pretty well known that bees from the Western European and Eastern European lineages are less diverse than African bees or those from the Middle East. So now we can talk about bees from San Diego. Bees from San Diego, pretty diverse when it comes to their genomic ancestry. Followed by bees from Mexico, also pretty diverse, more diverse than San Diego. Costa Rica and Panama, all pretty diverse. So this goes to show that admixed honeybees, Africanized honeybees, are more genetically diverse than pure lineages, which makes sense, right? If you're combining two distinct genetic lineages, you're going to end up with a lot of genetic diversity in the offspring. So that's why we have high genetic diversity in admixed Africanized honeybees. So that was all cool. You just got a whirlwind tour of honeybee genetics. And 6 years of my life I spent on a computer staring at my terminal and coding. And one day I decided I wanted to actually study the organism whose DNA like haunted me in my dreams. And so I decided to do an actual study of the actual organism, the honeybee. I wanted to put on a bee suit and go out into the field and actually see the creature that I was studying. So I decided to go and do a study of connecting genetics with behavior. And the behavior that I wanted to study was nest defense behavior, which is a behavior that I think most of us are pretty familiar with because we've been subject to that behavior probably at least once in our lifetime. So we maybe, how many people here have been stung? Okay, yeah, wow, that's a lot of people. Yes, we're all kind of familiar with getting stung. And defensiveness is a behavior that is pretty common and widespread throughout, the animal kingdom as well. Birds do it, ants do it, things like to protect themselves and protect their homes. So it was a behavior that I was really interested in studying. And actually, honeybees have evolved their defense behavior mostly in response to large mammals that like to steal their honey, like the honey badger here tearing away at a honeybee hive. And it was a behavior that was, really at the forefront of public consciousness because people have died from honeybee stings and honeybee attacks. So for those of you who are especially allergic in the crowd, please keep your EpiPen close at hand. And so I wanted to study nest defense. And how do you study nest defense? Very carefully, for sure. So I did this thing called the modified ratings test, where I would open up honeybee hives and remove some of the frames. And I would rate the tendency of the bees to run on the comb, to fly off the comb, to hit my veil when I was manipulating their frames. And I also would wave this little black patch on top of their frame and measure the number of stings that they deposited on that black patch. And I would also count the number of stings on my gloves after each inspection. And then I would also walk away from the hive for 25 to 50 meters and measure how many bees followed me, and to get a sense of how much they were willing to pursue me, to sting me. So at 25 and 50 meters, I would count the number of bees who were after me. And also, side note, how many of you have tried to park around UC San Diego? Okay, yes, it's horrible, right? It's horrible. But let me tell you, the best parking spot in UC San Diego is next to the honeybee hives. And I would always park my car. This is my car right here, next to the honeybee hives, a premier parking spot. And I dared anyone to come and give me a ticket. So pro tip, park next to the honeybee hives. So, and I rated the pursuit at various levels. So this is how I look when I go off doing my adventures. I have been stung hundreds of times at this point in my life. and I don't react at all. Thankfully, I didn't become allergic. I just became absolutely resistant. I put on my military grade pants and my suit, and I just know that I'm going to cry that day. So, and I open up the hives, and from May to November last year, I measured the best defensiveness in honeybee hives. And I actually measured defensiveness in two different sites. So we have our on-campus apiary, which is, these are hives that are managed, meaning that we treat them for pests and we requeen them with European queens. And so we kind of suspect that they're European honeybees, although we have never done genetics on them, so we don't know for sure. And I also tested hives at our off-campus unmanaged apiary, which these are hives that we've collected from across San Diego County. So they're feral, unmanaged. We think they're Africanized because no one has ever requeened them. So they're just kind of out doing their own thing. They're off campus because these are the hives that are a little bit more defensive. So we don't like that we keep them on campus. So we have kind of our unmanaged defensive hives off campus and our managed European hives on campus. And I just went ahead and I went to each site and I tested defensiveness in each site for 5 months. And this is a bar plot showing the number of stings on the flag. I'm just going to show you one metric of defensiveness because that would be a lot of graphs to show you of all the other metrics that I measured. But I think this one is the most telling. So and this is the raw data. I'm just actually going to show you trends in the data. So I plotted number of stings on flag over from May 2021 to November 2021. And what you can see here is that we have our managed hives. So these are, again, the European honeybees, and then our unmanaged hives, which are our Africanized unmanaged honeybees. And you can see how in May, they start off pretty low. The unmanaged Africanized hives are a little bit more defensive than our European hives. And then in July, they kind of both increase, but they remain similar. And then starting from August, you start seeing an increase in defensiveness in the honeybees in both sites, but more so in our unmanaged Africanized honeybees. And so as the year progressed, they just got more and more defensive, and I definitely noticed. So it was pretty cool to see a honeybee behavior change over months. And so I then did genetic analysis of these honeybees. I've already walked you through what these figures look like. So again, we have our ancestry panel here, and then let's show you our managed hives from the biological field station. And these honeybees are pretty European. They're mostly this yellow color, meaning they're mostly Eastern European. But what was surprising to us is that they actually have quite a bit of African ancestry, even though we requeen them pretty consistently. So that just goes to show that if for the beekeepers out here, which by the way, how many beekeepers are here today? Oh, okay, yeah, so this goes to show that if you're requeening it, if you're requeening your hives, meaning you're replacing the queen with a European queen in order to, for colony health, or you want to prevent Africanization in your hive, even if you're requeening pretty consistently, they're still getting some Africanized material into the hives, which was really interesting because we actively try to keep these honeybees pretty European. Let's move on to our feral honeybees, our honeybees from the off-campus apiary. So these bees are a lot more Africanized than our managed honeybees. So again, it shows that requeening bees does keep Africanization out of the hive, but they're, again, pretty similar to what I showed you in the San Diego sample. They're about, they have ancestry from all 4 lineages, mostly from the African and the Eastern European, but we have some genetic ancestry from the 2 other Western European and Middle Eastern clades. And so this is also really surprising because I have to tell you that San Diego sample that I showed you a couple slides ago, the ones that I collected And I showed you like the samples from San Diego, Mexico, Costa Rica, Panama. So I have to tell you that I collected those in 2015 and I didn't get to analyze them completely until 2 years ago. So that data was from 7 years ago. And so this data though is from last year. So I was kind of expecting the honeybees in San Diego to get a little bit more Africanized. I thought we were going to be seeing like 60–70% Africanization in San Diego honeybees, but the only increase in African ancestry by 2% over 7 years. So that goes to show that we're not really seeing changes in genetic ancestry in San Diego honeybees. What the genetic composition that we see in San Diego honeybees today is almost exactly what we saw 7 years ago, which is really interesting considering that in Texas, just a few years after Africanized honeybees reached Texas, 75% of honeybee genomes were African. So my next analyses are to tie genetics and behavior a little bit more. I want to see how exactly does ancestry tie into defensive behavior. I want to locate genes and different sections of chromosomes that correspond to to defensive behavior. And right now I'm currently working on some projects to tease apart and analyze the relationships between genetics and behavior. And I hope from this talk that you learned something about genetics and behavior in honeybees. I had a lot of fun talking about it. So I hope you had a lot of fun listening to what I've been doing for the last 7 years of my life. And I just want to also say that I hope you got some more appreciation for this really beloved, beautiful creature, the honeybee. I really fell in love with honeybees working with them. They're gorgeous, they're beautiful. They have so much to teach us. And I learn every single time I open up a honeybee hive. And I also want to say that they inspire me a lot. I do a lot of the artwork that you see in the slides, and I painted this mosaic because for me, like studying honeybee ancestry really was a gateway to studying ancestry in general and genetic admixture and the evolution of creatures and the biodiversity of our life. And so I had a lot of fun studying honeybees. And one last thing I want to talk about, because I know someone's going to talk about it, is this idea of save the bees. So I'm right there with you. I want to save the bees, but I also want to leave us with this idea that to save the bees, we really have to save, we have to be aware of like the intersectionality of our world. And when we talk about saving the bees, we also we want to save them from pesticides and pollution, but we also want to save our farm workers from pesticides and pollution. They are just as subject to those conditions as our honeybees. And if we're worried about honeybees, we should be very much also worried about our fellow humans. And if you are worried about pollution and pesticides and honeybees dying in droves, I definitely recommend to eat heirloom varieties, shop local, don't shop monocultures, try to shop local with organic local farmers who are cognizant of the things that honeybees need to survive. And also to support regenerative grass-fed farmers. Our world is changing, climate change is happening. So I think the best way to prevent climate change is to really put our money down where it should be put down and to shop local, organic, regenerative agriculture. Because revolution really begins in the garden, and what you eat is definitely where you should start to try to change the world. So I just want to leave you all with that. And I want to thank everyone who helped me make this project possible. All of my committee members, a bunch of friends, lab mates, and a big thank you to everyone here on this screen, especially my PI, Joshua Cohn, who has supported me so much through this journey. And with that, I would love to take any questions that you all might have. And thank you so much once again for being here and listening to my talk. Really appreciate it.